Vanakkam. Welcome to this course on biomedical instrumentation. In this week, we will look at biopotentials and recording of biopotentials. What causes biopotentials? How to record? What are the various recording techniques of biopotentials? In this video, we will look at origin of biopotentials. We will look at the excitable cell membrane and the biopotential maintained across the cell membrane. How is it achieved? Excitable cells maintain a steady potential difference across the cell membrane. How is it achieved? Concentration gradient and potential gradient, electric potential gradient, how are they maintained and how is a balance achieved between the two? And what is resting membrane potential of a excitable cell? Electrochemical gradient and electrochemical equilibrium. These topics are discussed in a bit more detail in my other course on neuroscience of human movements. I will ask the TAs to share with you the links in which detailed lectures on these topics are available, but this is a course on biomedical instrumentation, and so here we will briefly discuss this. So, what are biopotentials? These are potentials or electric voltages that are generated by ionic activity in excitable cells. What are excitable cells? Those cells that maintain steady potential difference across their plasma membrane are called excitable cells. These are neurons, muscles. Cardiac cell is also a type of muscle cell, that is, the heart is also a type of muscle cell. It is only one muscle of a type, it is a special muscle which has its own type. Cardiac muscle is a type of muscle and there is exactly one muscle of that type in the body. And these biopotentials are measured as potential differences between 2 points in, for our interest, usually between the inside of the cell and the outside of the cell. Why is this happening? Because the plasma membrane or the cell membrane is selectively permeable to specific substances and specific ions, and this creates an gradient, a concentration gradient, and because ions are also charged, it also leads to a potential, electric potential gradient. Here is an example of a case of a microelectrode kept outside the cell and another one that is kept inside the cell. This is a plasma membrane or an axon of a neuron, and you measure. Usually the resting membrane potential is about minus 70 millivolts. Note that this is not the case for all the cells in the body. It is a typical number for a typical neuron. So, remember, what is the cell membrane? The cell membrane essentially is a lipid bilayer that contains both hydrophobic and hydrophilic substances, and it contains different types of proteins. Some proteins span the entire plasma membrane. So, the plasma membrane has 2 layers. Note, some substances can diffuse through these, through the lipid bilayer. What are these substances? These are substances like carbon dioxide, oxygen, they can diffuse through the membrane. And some substances cannot diffuse through the membrane. So, it is selectively permeable. It permits some substances through its membrane and some it does not permit. Some proteins span the entire bilayer and these proteins are called as integral proteins, and some of these proteins also selectively allow specific ions through what are called as gating mechanisms. These are called as gated ion channels. In one of the videos in neuroscience of human movements, I would have described in detail the voltage gated sodium channel, but not sure how much time I can invest in this course on the topic, but do check that. So, these are gated channels. That means they allow ions of a specific species under certain conditions only. So, this is a very brief rough overview of the cell membrane. Typical concentrations of various substances and ions inside the cell and outside the cell are given here. Outside the cell, sodium is found in excess. Inside the cell, potassium is found in excess. So, outside of the cell or the extracellular fluid has 10x the amount of sodium. Inside of the cell has 30x the amount of potassium. There are also other ions that matter, calcium, chloride, and what is HCO3? Check what is HCO3. There are also others that matter. For the purpose of this discussion, we will keep our discussion confined to sodium, potassium, and perhaps calcium. But note, all these ions matter, not just one or two of them. Just because I am discussing one or two does not mean that they are the only ones that matter and the others do not matter. Every one of these matters, but I am only discussing some of the important ones. How is this concentration gradient maintained? Now, 14 is present inside and 140 units, that is milli equivalent per liter concentration is present outside. So, that means that there is a natural 10x concentration gradient. If there was a channel that would allow, if there was an integral protein that would allow passage of these sodium ions, then sodium would pass through this. But what is ensuring that this concentration gradient is maintained? Now, that is the question. How is this achieved? Through what are called as active transport. It turns out that there are different forms of transport. This is not a course on that topic. There are transport called passive transport, co-transport, active transport, osmosis and diffusion. So, and there are others like this. So, there are many different topics. One primary mechanism of interest is the active transport mechanism, also called as a sodium potassium pump. I have given an entire 1 hour lecture on how the sodium potassium pump was discovered by Jens Christian Skou in my course on classics in neuroscience. This is the other course. In that I have discussed in detail the great work of Jens Christian Skou in discovering the mechanisms of sodium potassium pump. We will discuss that very briefly here. So, this sodium potassium ATPase, also called as a sodium potassium pump, essentially has two conformational states. This is essentially a protein that has two stable states. I will keep the discussion a little less technical so that everybody can understand. So, this is a biomedical instrumentation course focusing on engineering students. So, I will keep the language a little less technical. So, there is a E1 state, which is the stable state in which the binding sites faces on the inside of the cell, and in this state this protein, which is the sodium potassium ATPase, has very high affinity for sodium, and when ATP is attached what it does is it undergoes a conformational change. It essentially hydrolyzes ATP. That means the third phosphate bond is usually broken, and it converts that third phosphate bond is the high energy bond. So, when that is broken, energy is released. When that energy is released, it causes a conformational change, leading to a situation in which it opens on the outside, and this side is the ECF or the extracellular fluid, and this side is the ICF or the intracellular fluid. Okay. Same for in this diagram, second diagram also. Now, in the E2 state, the binding sites face on the outside, and in this state, in this E2 state, the same protein, the same ATPase has high affinity for potassium and less affinity for sodium. How is this possible? Because this is a structural change, stable structural change that it is undergoing, and in this structural, it is having high affinity for potassium and very less affinity for sodium. So, what happens is sodium escapes from these to the outside and potassium enters inside. Let us revise this one moment. Sodium is found in high quantities outside. In this case, sodium is pumped from inside to the outside. That means, that is why it is called as a pump. That means there is an energy barrier that it has to cross, and once potassium attaches to the binding sites, it once again undergoes a conformational change, right. So, this is the E2 state, it once again undergoes a conformational change and opens on the inside, leading to E1 state again, and in this state it is again having high affinity for sodium. Note that 3 sodium ions are pumped from inside to outside, and 2 potassium ions are pumped from outside to inside. So, essentially what is happening? Essentially 3 cations are leaving the cell and 2 cations are entering the cell. Assume that these are not the same cations. That means that effectively one more cation has left the cell, making the cell a tad bit more negative. Is it not? Why? Because 3 positive ions have left, only 2 positive ions have come in. That means that the cell has become a bit more negative when compared with the outside, right, a tad bit more negative, which is why this process is called as an electrogenic process, that it creates a potential gradient. Note that this is happening against the concentration gradient. Potassium is also sent inside against the concentration gradient, because inside has 30 times the amount of potassium, outside has 10 times the amount of sodium. Sodium is sent outside against the concentration gradient. That is why it is called as a pump. If water is found on the ground level, you want tap water, running water, you pump water to an overhead tank. Why do you do that? Because if water is present in the overhead tank, if you open the tap, the water will flow naturally to the tap. Why will it flow? Because there is a potential difference, potential energy difference, is it not? Potential energy at the tank is higher than the potential energy at the tap. That is why. Why is it water from the ground not flowing and in the first floor tap? Because potential energy of the tap water is higher than the potential energy of the groundwater. So, to send water to the tap, you need to pump against the gradient. Yet, when I want to pour water from first floor to the ground floor, I just have to pour. Why? Because I am going in the direction of gravity. I am not going against gravity. When you have to send water up, you are going against gravitation. That means that you have to pump, you have to use pump, and that requires energy. You have to use either a diesel motor or an electric motor, something. You have to expend some energy. In this case also, one ATP molecule is used to send 3 sodium ions outside and 2 potassium ions inside, essentially an electrogenic process. This is called as the sodium potassium ATPase or the sodium potassium pump. This lecture I have given in detail in my other courses. Please do check for those who are interested in the physiology of how this works. This is beyond the scope of this course. Yes. Note that channel selective membranes. Now, what are these? See, remember, these are selective membranes, right? These are selective membranes means some can send only sodium inside, some send only potassium inside. Now, how are these done? It is like membership of a club. Let us say there is a club that only allows those above 18, some clubs allow only those above 21, for example. It maintains an atmosphere in which those above 18 only can enjoy, and it is not suitable for those below 18, for example, maybe. And there are these bouncers who are waiting outside, who are checking the ID cards. Are you 18? You are not 18, get out of here. So, there is a species specificity. You need to satisfy certain conditions to enter inside the club, otherwise you cannot enter. Likewise, if it is a sodium channel, only sodium can enter inside, potassium cannot enter. Likewise, there are some species specificity. What is more, you need to have membership of the club. If you don't have membership of the club, even if you are above 18, you are not allowed inside sometimes, depending on the club rules, for example. House rules, they will say, these are the rules. Why am I not allowed? My wish, it is my club, get out, right? Over and above this, maybe they do not allow informal clothing, for example. You have to be in certain types of clothing, right? If you are going in shorts and t-shirt, for example, some clubs may not allow. So, there are several different dimensions. It is not just age, it is membership, age, dress code, so many different dimensions alone. All of these are checked by the bouncers. So, maybe you can have different bouncers for checking different things, but the point is that there is a kind of selectivity, that ambience inside ensures that these kind of people only are mixed. Likewise, the cellular ambience is maintained by this selective, ion selective integral channel proteins, that is mentioned. It is an example, do not read too much into this example. It is the concept that is more important than the example. Okay. Resting membrane potential essentially is the potential difference that you find in excitable cells at rest. When there is no activation, what is the membrane potential that you find? That is called as the resting membrane potential. It is essentially established due to the interaction of multiple diffusion potentials of multiple ions. So, what are these various ions? What are these ions? Sodium, potassium, calcium, chloride. We discussed this. Ions will diffuse due to concentration gradient, but their concentration gradient alone is not the only factor. There is also a potential gradient. We will discuss that in a future segment, or maybe in this segment itself. How is this concentration gradient established? By active transport mechanisms. See, there are this active transport mechanism. So, I am having one terminal of a voltmeter inside and one on the outside, and I am measuring. At rest, I am finding that there is a, for example, a resting membrane potential of minus 70 millivolts. Note that these are examples. There are cases of different cells having resting membrane potentials between minus 40 millivolts and about minus 90 millivolts. So, this is the range of the resting membrane potentials. Okay. The typical is minus 70. So, when I keep on saying minus 70, do not say that this is the value for all the cells, that is not true. It varies by cell type, it varies between cells of the same type, so it can vary. At resting membrane potential, there is always some potassium that keeps on leaking. This potassium leaking, these channels that leak potassium are called as the leaky channels, leaky potassium channels. They keep on leaking potassium channels. They allow potassium to go outside So, the inside becomes a tad bit more negative, but then the sodium potassium ATPase keeps on pumping potassium inside. Why do this? It is inefficient. There are biochemical reasons why. So, the sodium potassium pump maintains the gradient through its active transport mechanism. So, so there is a concentration gradient, that is ion concentration exists across the cell membrane or the plasma membrane. Naturally, the movement, high school chemistry and physics, naturally the movement of the ions will be from a region of high concentration to a region of low concentration, and naturally that will happen. This is not going to require any sort of energy or a push or anything. This is a passive mechanism, pure diffusion. All that you need is a bouncer not stopping you, a channel that is open and allowing you to move, and then there will be a natural movement. So, it is a natural along the chemical gradient. Now, because ions are not charge free, ions have a charge depending on what type of ion it is. In this case, sodium and potassium we are discussing. They also carry a charge. When more number of ions are present on the outside or inside, there is an electrical potential gradient also that is created. This is the difference in charge or the voltage across the potential, across the plasma membrane or the cell membrane. Now, there are two different gradients. One is the concentration gradient, the other is the electrical gradient. And it turns out that positive ions are attracted to a side that is more negative, and negative ions are attracted to a side that is more positive, or rather positive ions are repelled by the side that is more positive, and negative ions are repelled by the side that is more negative. That is common sense, that is from our high school physics and chemistry. So, cations are attracted to the negative interior of the cell. Why? Because the inside of the cell is at minus 70 millivolts with respect to the outside. So, cations will naturally flow inside, not just due to concentration gradient, but also due to the potential gradient. So, the net driving force is essentially the algebraic sum, not just the sum, the algebraic sum of the actual membrane potential and the equilibrium potential for a given ion. Now, what is this equilibrium potential? We will have to see that. We will discuss this equilibrium potential in a future discussion. So, let us say an electrochemical equilibrium is achieved when you have multiple species of ions, for example, potassium, sodium, chloride and calcium. And it turns out that the resting membrane or the membrane potential, not necessarily the resting membrane potential, the membrane potential is essentially the weighted contribution, the weighted sum of the equilibrium potentials of each of these ions, weighted by the contribution that they make to the total conductance. So, each ion makes a contribution to the conductance. This is the sodium, this is the potassium conductance, this is the sodium conductance, this is the chloride conductance, this is the calcium conductance. The sum of all this, for example, is the total conductance. How much contribution are you making to the total conductance determines how much closer the membrane potential will be to that ion's electrochemical equilibrium potential. This equation is called as the chord conductance equation. Okay. It essentially describes the membrane potential as a function of the equilibrium potentials and the conductances and the relative conductances of the ions. Okay. And we could compute this for ideal cases and simple cases. I have done that in my other courses. Please do check that. This again is a little bit outside the scope of this course, but do check for your own knowledge and understanding. Another way to represent the same thing is the so called Goldman-Hodgkin-Katz equation or the GHK equation. two Nobel laureates, Hodgkin and Bernard Katz, are part of this, are part contributors to this equation. It turns out that instead of representing these as conductances, I could represent these as permeabilities, relative permeabilities. Now, PK, PNA are relative permeabilities. These are relative permeabilities, and then I could describe this as a function of the concentrations and permeabilities. In the previous case, it was equilibrium potentials and conductances. In this case, it is the concentrations and permeabilities. Okay. Now, K out and K in are concentration on the outside and concentration on the inside. Now, what is this electrochemical equilibrium? We know this from there is an electrical gradient and there is a chemical gradient or the concentration gradient, and I could essentially represent the force due to the concentration gradient using this equation, Fc is RT times ln of the concentration, and the force due to the electric gradient as F is Z times Fv, Faraday's constant, times the voltage V. Now, I have to do this for all the species, for a couple of species, for 2-3 species that is there in the system, and when I do that, I will essentially get Fc of 1 plus Fe of 1 as Fc of 2 plus Fe of 2 on the, for the two sides of a semi-permeable membrane. Now, this membrane is a semi-permeable membrane. Now, if there is an equilibrium that is reached, that is reached due to not because concentration has become equal on both sides, or not because potential has become equal on both sides, but because a balance between the concentration gradient and the electrical gradient has been achieved, or rather an electrochemical equilibrium has been achieved. In this state of equilibrium, there will be no net movement of charges. That does not mean that there is no movement, it just means that there is no net movement. What it means? What is the difference? If I say that there is no net movement of charges, that means if one positive ion moves from this side to outside, one moves from the in the opposite direction, so there is balance. That is what it means. It does not mean that there is no movement, it just means that the net movement is 0. It does not mean that the movement is 0. Alright. I could write this out for the two sides, on the outside and the inside, the two concentration, the two forces, and I could balance this. After some algebra and some simplification, I will essentially end up with this equation. This is called, this is the equilibrium potential in terms of the concentration and temperature, where R is the gas constant, T is the absolute temperature, and Z and F, Z is the charge of the ion or the valency of the ion, and F is the Faraday's constant. If I know these, in this I only have to know the inside concentration and the outside concentration for a given temperature. Say for example, I am interested in knowing this for the human body. A healthy, typical, normal human body when not in fever is having so much temperature. We discussed temperature measurements in the previous class. What is the temperature? I can substitute for that, and then I could compute, I could for example compute this constant, and then knowing the concentrations I could find what this VEQ is. What is this VEQ? This VEQ is of course called as the equilibrium potential or the Nernst potential. This is called as the equilibrium potential or Nernst potential. This equation is called as the Nernst equation. I could substitute for most of these constants. For example, this is at 310 K, T is absolute temperature. I could substitute for most of these constants and find out the values, and then make the solving of this problem easier. I have solved some of these problems in my previous courses on neuroscience of human movements, and I am not sure if I have done that on classics. I have done that in my previous courses. Please do check about this. With this, we come to the end of this video. In this video, we looked at biopotentials, origin of biopotentials, ionic concentration, concentration gradients, electrical gradient, resting membrane potential, and what is electrochemical gradient and how is it maintained, how is an electrochemical equilibrium achieved, and what are the various equations of interest. What are the various equations of interest? Card conductance equation, GHK Goldman Hodgkin Katz equation, most importantly the Nernst equation. And these are the equations that we looked at. And for more details, refer my other courses or refer some membrane physiology textbooks that will give you an idea of how this works. With this, we come to the end of this video. Thank you very much for your attention.